It's 150 slides, but mostly just like picture, picture, picture. Like it's.
We're going to be going through.
Yeah. There'll be anything else on the clocking line, so wait a minute. The side effect when you're doing all the similar stuff is that like anywhere just sit in the front row and I like come back. Has to be really good one for something like that. It's better for an example for a depends. For me, it was usually like the old retired professor. That wakes up the well, only one person. It's multi-task.
Hey, hello everyone. We're gonna get started. So I'm, for anybody who doesn't know who I am, I'm Katya. I'm a third-year PhD student in Lillian Aoki's lab, and I am hosting today's seminar speaker, Kaedan O'Brien. So Kaedan is an assistant professor in the anthropology department at SUNY Oneonta.
Oneonta.
I've been trying to say this all day. That's not happened. In New York, started in fall 2024. He got his master's and PhD in biological anthropology as a GRFP fellow at the University of Utah. And Kaedan has gotten several fellowships and prestigious awards, including the Lee Foundation's graduate research grants, and more recently the SUNY Seedfred Faculty Research Award and the Tübingen Prize for Early Prehistory and Quaternary Ecology. So I met Kaedan during my undergrad when I was a lab tech at the University of Utah in a geochemistry lab, showing him strontium isotope processing protocols. And shortly after that, he actually became my UROP mentor, where I completed a really substantial project for his dissertation. And, you know, I owe Kaedan a lot of thanks for being such an incredible mentor and showing me what being a good mentor would be. And yeah, working with him actually exposed me to paleoecology, which many of you know influences and is currently my work. So I know this talk is a little bit different than some of our previous I2 talks, so I'm really excited for you all to hear everything he's been working on. So let's thank Kaedan for being here.
Okay, thank you, Katya. So yeah, for those of you who aren't paleoecologists, this might be new, but I'm trying to make it as general as possible, understanding that y'all are coming from like 4 different departments here. So broadly today, we're going to be talking about paleo-environmental drivers of human evolution. In other words, how we can apply the skills of everything from evolution and ecology to environmental science to physical geography to better understanding, you know, the big questions of what makes us human. The question that I'm asking in my talk is simply, what are the paleo-environmental drivers of human evolution? This is one of the foundational questions in biological anthropology, arguably the question that we're supposed to be asking. And my goal is to place our hominin ancestors onto the landscapes in which they evolved. So here, for example, we have Ardipithecus, one of the earliest certain hominin species being shown in forested habitats in Ethiopia 4.4 million years ago. A lot of work goes into knowing whether to put them on top of a branch here, whether to put them into a forest or a woodland or a savanna or whatever. And we have much to say about the accuracy of this type of reconstruction and how we get there. So when we look at a paleo-environmental reconstruction, I'll be showing a lot of work in particular by fantastic Spanish paleoartist Mauricio Antón today. But take, for example, this Homo ergaster in Kenya at about 2 million years ago, including before our formation. You see the hominins carrying away some elephant meat over here. You see the hyenas, you see other things going on. Everything in this image is something that derives from paleo-environmental reconstruction, from people studying everything from environmental DNA to studying pollen and palynology, to studying the large vertebrate fossil record, to studying the behavior and stone tools of the hominins themselves. All of this comes together to piece together something about human evolution. In this case, lots and lots of things. The issue is that all of this is assumptions about past hominin behavior, and this allows us to begin answering questions about what makes us human and who we are. But tough anthropological questions like that require improved paleoecological methods. And this moves beyond studying the context that we evolved in, just the green background of human evolution, but taking it a step further into understanding the drivers of human evolution. It's one thing to know just like what the background was. It's more important to know why did that shape us into the strange species we are today. And, oh, okay. So today I'm broadly going to be asking 3 research questions, each at a different stage in human evolution. So we're starting not too long ago, just in the past 100,000 years, asking the question, how did African animals respond to past seasonality? We'll then move on to a couple million years ago, asking, did Homo and Paranthropus, 2 hominin genera, live in different habitats? And we're actually going to end more like 4 or 5 million years ago, asking, did hominins evolve in savannas or forests? And these are all kind of hypothetical questions, and I'm by no means solving any of these things, putting it in any of this today. The goal is just to show how we do this research. I'm going to spend the biggest amount of time focused on this first question, how did African animals respond to past seasonality, because that has multiple components. The latter 2 are just to give more deep time examples of some of the ways to apply these methods deeper and deeper into the paleontological past. Okay. Throughout the course of today, I'll discuss findings of prehistoric migration in Kenya, evidence that our hominin ancestors lived in different environments from one another, and introduce a new project that I'm just kind of starting now for evaluating whether hominins evolved in forests or savannas. But let's start with question 1, and again, this is where I'm spending most of my time today. So let's jump over to East Africa to the greater Serengeti ecosystem. Everyone who's ever watched a nature documentary, usually voiced by David Attenborough or some other British ecologist, knows that this is the largest terrestrial migration in the world, and the Serengeti migration of wildebeest, zebras, and Thompson gazelles is among the best understood natural phenomena in the world, we think. The issue is we literally know nothing about it prior to about 200 years ago. We have no ecological or cultural knowledge of how old this migration is, despite, again, having that David Attenborough voice in the back of my head always telling me, well, this is timeless. This has always been this way. This is nature at its best. We know that East Africa hasn't always looked the way it does today. So whereas today, this grassy ecosystem on the border of Tanzania and Kenya, it's relatively small, greater Serengeti ecosystem that the wildebeest, zebras, and gazelles migrate through. If we go back in time, just 12,000 years, we see that these grasslands are expanded far and wide, about from Uganda all the way to the Indian Ocean coast. And in fact, even modern Lake Victoria would have been a dry, grassy lake bed at the time. So if we want to begin understanding what this ecosystem was like in the past and how it functioned, we start by looking at the species that are occupying the region today. So today, most of the critters hanging out in the Serengeti ecosystem are non-migrants. They're species that either have stable, very localized home ranges or move around very little, like most of the animals on this slide: impalas, topi, orbi, buffalo, all of the small animals, and so on. That said, there's a really outsized impact on the ecosystem of these 3 species: plain zebras, blue wildebeest, and Thompson's gazelles, which have a greater biomass than everything on the last slide combined, and 70 other species, just because by migrating, they're able to sustain large population sizes. We obviously aren't dealing with squishy animals covered in fur and flesh and things like that. We're dealing with a lot of busted up horns, leg bones, teeth, and things like that. So this is what the fossil record tells us about the Serengeti ecosystem in the past. That said, we can begin to piece together a lot of details about what this ecosystem was like. First of all, we know that pretty much all species that are still alive in the Serengeti were around in the last glacial period. So the period of time from roughly 100,000 to 12,000 years ago, the last ice age of Eastern Africa. We had basically the entire set of modern animals, but we also had a few others that are now either extinct or extraliminal. So things like the extinct roosting oryx, a giant bellowing wildebeest, was actually the most common animal in this ecosystem. They're completely extinct now. Whereas others, like red zebras and white rhinos, are still alive, in both cases very critically endangered, but they no longer live in that specific ecosystem. They live in drier habitats in other parts of Africa. The first thing I want to ask is, how do we study ancient migrations? How can we piece together what a migratory system was like in ancient Africa? And we do this in a very convoluted way that seems a little bit like magic, and this is actually where my work with Katya comes in. So we do isotope sampling of the side of teeth for the element strontium. So teeth grow kind of like like tree rings, but in this case from top to bottom, from crown to root. So here, for example, this is my doodle of a buffalo molar here. And we have the enamel surface of the tooth. Everything above the root is enamel on the outside. And hypothetically, we could drill with a little Dremel bits of enamel powder from the outside of that tooth. It may be 2 to 3 millimeter increments. And since that tooth takes about 2 years to grow, if we do this, say 20 times, we're getting a roughly monthly or bi-monthly signal of something the animal was doing being recorded in that isotope powder. Now, depending on what element we look at, we might study carbon for diet, or oxygen for water intake, or nitrogen for trophic level. In this case, let's talk about strontium, because strontium is specifically useful in studying movement. And that's because strontium varies a lot across the landscape. So here, for example, is a great study by one of my colleagues, Janssen, from 2020, showing just how variable strontium is just across the borderlands of Kenya and Tanzania. We see these different colors on this chart correspond to differences in the third decimal in strontium. And if we zoom in our specific area, you can see a huge amount of variation across our study sites of Kenya Hills, Kibogo, Rusinga, and Kurubu. Now, importantly, again, note that the variation is at the third decimal here. Our instrumentation at the University of Utah, where all of this was processed, is accurate about to the fifth decimal. Meaning that if an animal moves from location to location, we should be able to really readily pick that up in their dental signature. Here, for example, is 2 test runs that Katya and I did 4 years ago now of the same enamel from the teeth, just to make sure we're getting the exact same signals each time. As you see in the runs 1 and run 2, we're getting accuracy down to that fifth decimal. So I set out to answer 3 objectives for this first part. First, I want to evaluate migratory behavior in one species, started with the most common species, roosting oryx atopocranian. We then expand this to all other common bovid and equid species, so all antelopes, buffaloes, and horses. And finally, we evaluate how migration and other seasonal behaviors correlate in both the past and present. So we start with roosting oryx atopocranian for a few reasons. First of all, it's the most common large herbivore in this ecosystem. It's also found in mass death assemblages, showing that it was a key prey item for human foragers during this time period. We were hunting them. And third, it has the ecomorphology you would expect for a grassland migrant specialist. Not only does it have extremely long legs with really narrow joints, which is what you would get in a species that is running and walking in straight lines. That's something you would expect in a migratory species. It also has odd head morphology consistent with something that was communicating over miles. So roosting oryx is one of the few mammals that actually is conversant with hadrosaur dinosaurs in having this trumpet pressed on its forehead that would allow it to bellow for something like 10 to 20 kilometers and be heard at these low infrasonic frequencies, kind of similar to actually how elephants communicate with one another. But this is a wildebeest doing this. So long story short, if I had to pick one of these species that's probably migrating in the past, it's this one. So this is a good test study. So one of my summers in Kenya, this is 2022, I spent sampling every tooth I could find from the site of a bunch of different species, starting with roosting oryx. So I did this all with my fantastic Kenyan undergrad from the State University, Lillian Ashioya. And to begin with, let's talk about these 4 roosting oryx teeth. So these are all second and third molars of this species of giant wildebeest. You can see my sampling lines there. This all gets processed and brought back to the University of Utah in the fall of 2022, where I worked with Katya. in processing all of these samples, which took about 1.5 years in total to get through all 800 specimens. And we get our results. Now, before I actually pop the results up, I just want to explain the axes here so you know what you're looking at instead of just a squiggly line. So on the x-axis, we have the millimeters from the cervix. This is the base of the tooth. So for example, 40 here means it is 40 millimeters above the root of the tooth. So we're tracking the tooth from top to bottom here. And then on the y-axis, we have strontium 87 over 86, which again plots onto that colorful Christmas color map that I showed earlier. Hypothetically, if an animal is not moving, it should have a completely stable strontium signature. If it is moving, it should wiggle up and down, or at least in one direction or another. I've done the honors here of, to really simplify this for visualization purposes. In gray, I've highlighted the total local strontium variation within 10 kilometers of the depth site of where this particular tooth is from. Now, no, I can show this for all 80 specimens. Not going to do that for you guys. I'm going to keep things simple and just start with some individuals and then broaden out. What do we see? Our roosting oryx were coming and going from the area where they died. In fact, they were probably born outside of the area, migrated in, migrated back out, and migrated back in before they died. This is consistent with an annual migratory system, similar to what modern wildebeest do in the Great Barrier ecosystem. This is such a beautiful sine curve here. This is like what I dreamed of getting. Most do not look this pretty. When we look at the other roosting oryx specimens, every single one in our analysis looks like these four, in that they either come and go from the local region where they died, or that they were never developing in that region in the first place, and they simply migrated to it at some point before death. In other words, none of these individuals had localized lives. So we can conclude that roosting oryx was migratory during the last glacial period. And in fact, this is the first evidence of prehistoric migration in Eastern Africa. Using strontium for this is now kind of common in things like reindeer in Europe, mammoths in Alaska and the Yukon. There's been one study of blue antelope in South Africa. This is the second strontium study of this type in the African continent, and the first that found results of positive migration. And this is now published in Frontiers in Environmental Archaeology. Okay, that's just one species though. That's just a setup for what is clearly a bigger project. What about all the other critters in this ecosystem? What about the elands and impalas and vespa, the other wildebeest species, reedbuck, zebras, and so on? I sampled the 18 most common species in the assemblage, and these are the strontium results for them. So given that there's so much data here, I don't want to go into detail of every single one. Instead, to make this visually more appealing, instead of looking at wiggles, I've simply made them box and whiskers plots, and again, highlighted in gray the local strontium signature. And what we see is that most animals have very little variation, and for the most part, they fall within the local variation. These four immediately stand out, though, as having different values and higher amplitude. This is specifically Megalotragus and roosting oryx, two extinct wildebeest taxa. The issue is that there might be a bias here of sampling effort. I got a little concerned when I got these results because these happen to be two species that have really tall teeth, and I was able to sample more times than the others. So I wanted to make sure I wasn't just getting a false positive. So this, the solution is to do a simple permutation analysis and standardize the sampling sizes for all of them. And I decided to take things a step further and set up some clear criteria for who I'm defining as a migrant and not. So to define a fossil animal as a migrant, I set up three really strict criteria. They have to fall outside the local strontium range. They need to overlap in their standard deviations of variation with the roosting oryx, a known migrant from that locality. And they need more variation than the small bobids like this Orabi here that we know aren't migrating because their legs literally couldn't carry them that far. And the exact opposite needs to be true to define an animal as a non-migrant. They need to not move and be within the variation of the small local bobids. Anything that doesn't meet all three of these criteria, either positively or negatively, remains unclassified. When we do that, we actually see the exact same results. These two species are the only ones that meet all of our criteria. So only Brucingorix and Megalotragus are confirmed as migrants. Of the rest, 63 out of 71 are non-migrants. The remaining 8 remain unclassified because they have mixed criteria. Importantly, that includes 21 out of 25 of our blue wildebeest, Thompson's gazelles, and plain zebras. The rest meet 1 criteria for migration and 2 for not migrating. In other words, our modern migratory species that you would go see on safari in the Serengeti today weren't migrating as recently as the last glacial period. And this is now published in Nature, Ecology, and Evolution. So this has some really big implications. First of all, the most abundant herbivores were migratory in the early last glacial period, including Brucingorix, and this is Megalotragus, our giant kind of ox-sized wildebeest. Migration in these species may be linked to their extinction. Most of our sites are in places that are now covered by Lake Victoria, but wouldn't have been at that time. They go extinct within 1,000 years of when Lake Victoria fills back up, cutting off their migration routes. That might not be a coincidence. Importantly, migration is not happening among the extant species that migrate. They seem to have developed it more recently. This is a little bit of speculation, though I will say this is supported by some preliminary results by a colleague. It actually seems that canyon migrations of wildebeest and other species only emerged in the Holocene once you have cattle brought in from Ethiopia, maybe in the past 5,000 years. If that makes you feel uncomfortable, good. It makes me feel uncomfortable too, that one of the biggest natural phenomena in the world may actually be anthropogenic. So I set out to answer one final objective with this first project: evaluating how migration and other seasonal behaviors correlate in both the past and present. So for this, I asked 3 questions. How has herbivore diet changed since the last glacial period? How is the extent, or what was the extent of diet switching in the last glacial period relative to today? And finally, how do these different seasonal behaviors relate to grass dependence in the last glacial period versus presence? How are migration and diet related, in other words? And this is based on a really cool paper by one of my biology collaborators, Joel Abraham, who's a Princeton PhD, well, now defending his PhD last month. And he found that the more grass you eat, the more likely you are to migrate. And the more mixed feeding you are, the closer you are to 50% grass in your diet, ranging from grass to browse, leafy vegetation, the more likely you are to switch diets. Both of these things kind of make intuitive sense. I wanted to test whether they hold true in deeper time. And I do this using delta 13C, the simple carbon isotope used to tell tropical grasses versus all other plant vegetation, for the most part, in tropical Africa. It's a bit more complicated than that, but I'm happy to answer questions about it later. So first, asking how has herbivore diet changed since the last glacial period? We literally just can compare fossil species in their diet to their modern counterparts. Here, for example, we have an impala. This is what its delta 13C is per time, suggesting that it is a mixed grazer. Here we can summarize that for all of those same teeth that we also sampled for strontium. It's all the same individuals. I have this divided, so in yellow, I have at our 4 sites. In yellow, I have grazing, grass and sedge diets. In dark green, I have browsing, leaf and fruit diets. And in lime green, I have mixed grass and browse diets. What we immediately see is basically everything is grazing during the last glacial period, which is completely different to today, where the whole spectrum is represented. So high grazing signal in nearly all species show that there were clearly diet shifts across the Pleistocene-Holocene transition, which again makes sense if you keep in mind that all of East Africa used to be a giant mega Serengeti at the time. Of course, they were all eating grass. That's the easiest part of this story. It gets more complicated after this. What was the extent of diet switching in the last glacial period relative to today? If we look throughout a tooth, just because they're a grazer, are they always a grazer, or do they switch diet throughout the year? So I want to compare to the modern data of modern herbivores in East Africa, looking at grass dependence, diet switching, and coding 1 or 0 for whether they migrate or not. Starting with diet switching, we see that in almost every case, with one exception, it's a really important exception, our fossil herbivores are diet switching much, much more than their modern counterparts. A modern wildebeest eats 99% grass. Winter, spring, summer, fall, does not matter. They eat grass. Go back in time 20,000 years, and that is not true. Some parts of the year, they're eating 100% grass. Other times, it might be as little as 80%. So we have much higher diet switching in the last glacial period. So we're building up our data sets now to ask our really big question, which is, how do all these seasonal behaviors relate to grass dependence in the last glacial period versus present? So again, in the past, if you eat grass, you always eat grass. If you browse, you always browse. If you incorporate both, you always incorporate both at different parts of the year. Note that there's nothing in this category here. There's nothing that splits its time between grazing and browsing consistently throughout the year. There are a lot of things that graze often during the rainy seasons and browse during the dry seasons. We go back in time to the last glacial period, you see that this relationship completely disappears. We actually see not really much of a relationship at all. I will say this is actually significant, that the more grass you eat, the more you diet switch. Now, obviously, if you 100% are a grazer, you can't diet switch, but right up until that point, you're the most likely to be diet switching. Note here, for example, our highest diet switchers at 38% of their diet, 38% diet switching, are some Brucing Oryx individuals that are consuming something like 88% grass on average throughout the year. What that tells us is that some parts of the year they're eating 100% grass, other times it's less than 50%. So, diet switching was a grazing strategy in the last glacial period. What about migration? So migration, by subset our data set just down to modern African herbivores, we see that basically if you browse, you don't migrate. If you graze, you are more likely to migrate. A simple GLM model here. In the last glacial period, again, that correlation falls apart. Basically, nobody migrates other than Brucing Oryx and Megalotragus, neither of which is a full grazer. They're both these mixed grazer species, suggesting that this was a mixed grazing strategy in the last glacial period. Now, we compared this in a bunch of different ways, but this can all be seen in better detail if you just read the paper in Ecology Letters, non-analog behavior of Eastern African herbivore communities during the last glacial period. So, our biggest conclusion is that behavioral responses to seasonality were flipped in the last glacial period. Some implications of this. Palatable grasses were still a limiting factor even in the last glacial period. Even if you live in an open Serengeti that goes from Uganda to the Indian Ocean, maybe as far as the Congo to the Indian Ocean, it doesn't matter if the grass dries out and is low protein, you need to supplement your diet with other foods in a way that even modern wildebeest don't have to do. Behavior was therefore non-analog in the past, despite among our 18 species, 14 being extant species you could go see on a safari right now in Tanzania. So it's all the same species, the same subspecies even, if morphology is confirming that, yet they're behaviorally completely switching their behavior. In other words, we can have a Western white-bearded wildebeest, the subspecies that is 100% migratory, but we go back in time 12,000 years and they have a completely different ecology where they diet switch to respond to seasonal changes instead of migrating. Very, very cool and says a lot about behavioral plasticity. So, to summarize this first big chunk of the talk, we have the first evidence of prehistoric migration in Africa. The migration is limited to extinct wildebeest and absent in the extant migratory species. And we see really odd correlations between grass consumption, diet switching, and migration, suggesting that some zoological assumptions that we make and read about in behavioral ecology books don't really pass muster if we extend things into even just a slightly different carbon dioxide regime in East Africa. Now, this may be a coincidence, but what the archaeologists are taking from this is that this may tell us something about human behavior. Because as these migratory giants are going extinct, this is precisely when humans switch from what is known as Middle Stone Age to later Stone Age technology. Knowing that I'm not talking to a group of anthropologists, I'll broadly say they're switching from relatively simple tools to more complex blades, arrowheads, things like that. One could argue that this is because their prey choices are changing substantially. You're no longer mass killing Brucing Oryx, you might be shooting at rabbits. So, on to the next part of the talk, much more brief, but talking about applying some of these paleoecological skills to deeper time, in this case, studying Homo versus Paranthropus. How do spatial and temporal scales of reconstruction impact our interpretations of human evolution? So this is a huge issue in anthropology. So scale and environmental reconstruction go hand in hand because depending on what proxy you're looking at in paleoecology, you might be reconstructing something very different. Some sets of data might tell you about the localized habitat, that square meter of land. Other things might tell you about the whole basin or an entire region or the globe or something like that. The point is we need to match our processes and questions with the scale of our data. So a case study in this is the Kubifora Formation, where I've been working since 2017 in northern Canada. This is Kubifora, beautiful, beautiful landscape, very similar looking to the Badlands of South Dakota, but up the temperature to about 120 degrees all summer. So I have past work in this basin that studied some of the spatial variation and temporal variation. If you look at different scales, you see different stories happening in this formation, going all the way back to my first paper ever in my master's. I decided, though, that I wanted to apply this to studying human evolution more directly. Homo and Paranthropus, two hominins, human ancestors that lived very different lives. Now, both of these genera are found in the Kubifora Formation. On the left, I'm holding a Homo rudolfensis premolar. On the right, I have an unerupted M1 of Paranthropus boisei. So human evolution isn't a nice linear story. Instead, we've always had diversity within the human lineage, really until 39,000 years ago. Whereas early Homo, at this point, we're dealing with Homo habilis, Homo ergaster, have a relatively large brain, small teeth, and an omnivorous diet. Paranthropus boisei are defined by having a small brain, enormous jaws and teeth, and being herbivorous grazers. You can really look at these as being sort of the black bear generalist and the panda herbivore specialist, but applied to humans in this case. Now, we've long known that they were sympatric and interacted, as we see by this lovely paleo art from the 1970s of them going to war with one another. We know that they lived at the same places at the same time. How, though? How do you have sympatric great ape species so closely related to us coexisting? We know that they were dietarily niche partitioning based on multiple proxies I won't get into today, but I wanted to investigate whether they were environmentally niche partitioning too. So I asked, did Homo and Paranthropus live in different habitats from one another? And my hypothesis, really parroting other hypotheses that have been proposed since really 1976 by Richard Leakey, are that early Homo was an environmental generalist, while Paranthropus was an environmental specialist. Shipman and Harris write about this in 1988. Elizabeth Verba, same thing, 1988. There have been some studies, really one by Bernard Wood and David Strait in 2004, suggesting that there isn't really evidence of this, but I don't think they're really testing at relevant scales yet. So I set out to answer this just as kind of a pre-PhD project. I decided to test this using the faunal record, the large herbivore record. And the Kubifora Formation is a great place to test this because we have high spatiotemporal resolution and high sample sizes. If we can get this right, we can solve a 50-year-old mystery and one of the biggest questions in paleoanthropology. So we've already been shown by my master's thesis, as I talked about, that the different regions of the Kubifora Formation have different ecologies at different times. What if we break things down even further? So I break down the Kubifora Formation into a bunch of different units, both spatial and temporal, that I basically treat as assemblages. These are assemblages of hominins and their associated fauna. This is them all mapped out. Note that they date to a few different time periods, all broadly from 2 million to 1.4 million years ago. And the objective is to evaluate the faunal associations of Paranthropus and early Homo in the context of the specialist versus generalist debate. Again, if you're an anthropologist, this is a debate that's been going on since the mid-1970s. If Paranthropus is an environmental specialist relative to early Homo, its fossil occurrences should be associated with faunal assemblages indicating a narrower range of environments, whereas Homo should be associated with all kinds of different faunal assemblages. And we can test this in a lot of ways. I test it using specifically the family Bovidae, the antelopes and wild cattle. And this is mostly because they have really established environmental associations at the tribe level. So tribe being the mammalian level between genus and family, ranging from wet to dry and open to closed. So take, for example, the wildebeest tribe, the Alsalfini, are really associated with open, dry grasslands. Whereas, for example, the kudu, the Trigellofini, are associated with dry woodlands. Redundancy on the top right are strongly associated with floodplains. So we know that there's really strong environmental associations with these different tribes. And we can plot this out using simple correspondence analysis here. If we look at all of the tribes shown, all the Baba tribes in orange, we see that they plot out very differently in their association with one another in modern African game parks, shown in the black triangles. For example, Kruger National Park in South Africa is dominated by woodland ecosystems and the tribe Apis cerutini, the impalas, whereas Impholosi, also in Southern Africa, is more intermediate and is dominated by Cape buffalo. What we see there also in purple, these are all of our 34 assemblages, our 34 assemblages, and we see that they also span quite a range, though not getting into the full-on woodlands down here. So we could hypothesize that maybe we'll find Homo in the forests, in the woodlands, in the grasslands, in the swamps, everywhere, but maybe we'll only find Paranthropus in one of these assemblages. If this were true, this would confirm our specialist versus generalist hypothesis. What do we find? Kind of that. We see that Paranthropus has a narrower ecospace that it occupies relative to Homo. Paranthropus is found in this convex hall of sites that is a bit smaller than that of Homo. This despite Paranthropus actually having a larger sample size. So if anything, based on randomness alone, you would expect it to be bigger. That said, I don't trust the code enough to just base things off of that alone. So instead, I established a null model where we randomly assign hominins and other species to different assemblages and see what's actually statistically significant. When we do that, we find that this convex hull of Paranthropus is statistically smaller than random. So in the bars, this is our random hull size, it's the size of that polygon of association of ecospace. In our correspondence analysis, Paranthropus versus Homo based on the sample size. In the gray, I have a 95% confidence interval. You see that whereas Homo has a very big convex hull and well within a random assortment of Homo at the different sites. If you just place them randomly, dump them out of a plane, that's exactly where they would end up. Paranthropus has a significantly smaller convex hull size than random, suggesting they are in fact ecological specialists. So early Homo is an environmental generalist and Paranthropus is a specialist, kind of solving this 50-year-old mystery. So again, this is coming back to this big question that environmental variation can only be understood at appropriate scales. This question had been eluding us for 50 years because we weren't really looking at it in the right way or using the right kinds of data. Finally, for the last very brief part of the talk, I want to talk about a new direction that I'm moving into, really talking about one of the biggest chips on my shoulder as a paleoecologist, trying to answer the question, did hominins evolve in savannas or forests? So, the savanna hypothesis is one of the biggest hypotheses, theories, depending on who you ask, of human evolution that dates all the way back to Charles Darwin. So the original version of the savanna hypothesis is that essentially some chimp-like or gorilla-like ape came down out of the rainforest and walked upright into the savanna. Obviously, we're talking about, maybe this is a million years of time. This is one individual pioneer, but the story kind of stays the same. You have these rainforest apes evolving into these savanna-dwelling bipeds that eventually become us. And if you ask Raymond Dart and Charles Darwin, this is thought to explain everything about us. Bipedalism, our big brains, hunting, tool use, all of that. And anthropologists often just accept the savanna hypothesis as fact without question, but isn't that a terrible idea for literally what should be one of the biggest questions on the planet, what made us human? So I think like let's actually put some rigor to this. Let's put some math behind this. Let's actually see what the evidence shows us. So I personally have a few big problems with the savanna hypothesis. First of all, being apes have been living in grassy ecosystems for something like 21 billion years, and yet it's just our particular brand of apes that have become bipedal in the past 7 million years. Second, the shift from forest to savannas was gradual. When humans split from chimpanzees roughly 5 to 8 million years ago, nothing really substantial was happening. In fact, it was a pretty stable time for the climate. Bigger pulses were happening at 20 million years ago and 10 million years ago and 4 million years ago. So why at 7 million years were our type of apes forced from the forest into the savanna? Finally, why just humans? Lots of other animals live in savannas and they don't become bipedal. Why do we have bipedal lions or baboons? No one really is asking that. So I'm really skeptical of the savanna hypothesis and think we need to see more evidence that humans were for sure not just continuing to live in forests until much more recently. And the place that I'm going to be testing this is the Cimarron Formation. So here we're looking at the Great Rift, the East African Rift System of Eastern Africa, and specifically Lake Baringo and the Tugan Hills just west of them in West Central Kenya. This is the traditional homelands of the Tugan and increasingly Pokot peoples. So the Cimarron Formation is just one of about 17 million years of sediments that we have in the Cimarron, or in the broader Tugan Hills sequence. You can literally spend a day walking from the north to south end of this mountain range and walk through 17 million years of deposits. This is actually the broadest Cenozoic stratigraphic section in all of Eastern Africa. So you can literally watch. I mean, at this point in the sequence, we haven't even split from orangutans and gibbons yet. By the end of it, we have Homo sapiens fossils. So the Cimarron specifically dates from 5.3 to 1.6 million years ago and covers really most of the key events that we think about in hominin evolution. There are 240 fossiliferous sites, so fossil-bearing sites, only some of which have hominins. But importantly, they're kind of just sitting on shelves. A lot of these were collected in the 80s and 90s, some even before that. They've been identified maybe to family or genus, but nothing has really been done with them since then, which is very weird because we have a lot of hominins at these sites. We have Ardipithecus, Australopiths, Homo, and Paranthropus all coming from the Cimarron Formation. So I want to do something with this. And I think the Cimarron is the best place in the world to test the savanna hypothesis because we have so much hominin diversity and turnover through time. So Ardipithecus ramidus is mostly known from Ethiopia, but we do actually have a mandible of Ardipithecus ramidus called the Tabarin mandible coming from the Cimarron Formation. And depending on who you ask, it is either a tropical rainforest or an open savanna like the Serengeti. Surely we can sort that out. The issue is that different proxies are telling us really opposite information. If you're looking at the animal fossils, you're seeing tons and tons of rainforest dwelling fossils. We have green peacocks, a species that today, the sister species, I should say, to green peacocks, a species that today lives mostly in Indonesia and mainland parts of like the Malaysian Peninsula. Dense rainforest, even more so than the more common blue peacock. One of our most common large mammals are water shepherds, small mouse deer-like animals that live only in forested riverways in places like the Congo today. So if you only ever look at that data, you'd say, oh, they lived in a rainforest. Stop. Next question. But if you look at the pollen samples, it's dominated by grass. So depending on what paleoenvironmental proxy we're looking at, we're getting different results. And I think that basically, if the right is true, then this would confirm the savanna hypothesis. If the left is true, we need to start over and go back to the drawing board. Realistically, we might end up somewhere kind of in the middle, and I argue that this is simply a matter of scale. The landscape might have looked more like this. This is actually a different part of the rift system today, where we see, for example, a tropical corridor with lots of fig trees and vines in what you would still call a savanna biome. If we're dealing with this, we might have tons of grass pollen, but a bunch of rainforest-dwelling animals running around. So my hypothesis that I'm only beginning to test now is simply that, yeah, hominins were living in the forested corridors of a savanna landscape. We were living in the savanna, but not the open part of it that Raymond Dart and Charles Darwin would have pictured. We were still swinging in the trees, just like many monkeys are living along riverways, cutting through the deserts even. And we're collecting a team right now to study this in a lot of different ways because environmental change is affecting the Baringo Basin during this time in a lot of different global to regional to local scales. So we have different factors, global abiotic factors like solar insolation, glaciation, starting up and atmospheric CO2 changing. We also have regional abiotic factors like movement of the intertropical convergence zone, uplift and rifting of the East African Rift system, but then also local abiotic factors like paleolake dynamics and fire. Finally, we have our biotic factors to look into. We have global biotic factors, notably grassland expansion at this time, but then also the local biotic factors, animal community change, dietary shifts, and so on. This is my role in this project, is looking at things like faunal remains themselves, the fossil antelopes and zebras and so on, and also the stable isotopes of their teeth. So I've started a pilot study, and this is kind of the way my research is moving over the next few years. I'm just looking at 435 new fossil specimens collected in the 2022 field year. In that alone, I've already found 2 new antelope species, just showing how much potential there is if people just take the time to identify things like they could. I don't know if I should show this, but it's my data, so I don't care. This is a new species of antelope, distantly related to wildebeest, next to its closest relative that is twice the size of it. So there's a lot of work to be done here if we just get the brains involved and get more people involved with working in the East African Rift system. So to summarize that, testing the validity of the savanna hypothesis requires understanding paleoenvironments across multiple scales. We can't just be happy with, oh, it's a forest or it's a savanna. Well, at what scale? Are we talking about a habitat or are we talking about a biome or a continent? What are we dealing with here? Then we can begin to answer these big picture questions in anthropology. So, through this talk, I've demonstrated evidence of prehistoric migration in Kenya using stable isotopes, evidence that our hominin ancestors lived in different environments using quantitative analysis and correspondence analysis, and finally discussed a new project that I'm starting for testing whether humans evolved in forests or savannas. And really, my take-home message is simply that improving our understandings of the past will make images like this more accurate and help us truly know how and why humans evolved. We shouldn't be happy with the stories that we were taught in Intro to Bioanth. We shouldn't be happy with the stories we were taught in high school biology. These are open questions, and we need a lot of people working on these types of questions using a bunch of different proxies, whether paleozoological, paleobotanical, geological, biological, genetic, and so on. And we can hopefully begin to understand more and more about what made us the strange apes that we are today. I could go on forever, but I'll end there and open things up for questions. Thank you. If anyone has questions, yeah, I'll start. Yeah.
You may have mentioned this and I just missed it, but going all the way back to your migratory strontium analysis, do you see that same sort of sine wave pattern in the modern animals? Okay.
Well, I mean, I purposely started that because that's like the prettiest one that illustrates the point. It all depends on what the nature of the landscape is. If they're like in that case, if they're literally moving in a circle between a low and a high region, it will always look like that. That's not always how animals move. Take elephants, for example. Elephants don't always migrate in circles. They like to just kind of do this on the landscape, and they're just all over the place. So it really depends on what pack they're on. Elands are also a nightmare. Male eland often migrate 100 kilometers or more once they reach sexual maturity, and their strontium just goes all over the place. So what I'm showing here is a very, very, very oversimplified version, but I'm happy to talk about the quantitative methods in more detail if you want to know. Yeah. Any other questions? Yeah.
So how does the strontium that variation change over the course of the time scales that you're thinking about? Do we know that has been pretty stable?
Yeah, great question. So strontium basically is stable as with the volcanic formation of the rocks. So strontium-87 is radiogenic, so it's created from rubidium breaking down, or rubidium-87 breaking down into strontium-87. And then neither 87 or 86 are radioactive, so they're stable after that point.
Yeah.
So basically, any rocks, so the rocks are based on just like your geologic map. They break down into the soil, so as long as those soils are derived from the underlying bedrock, you should be fine. Now, some people do strontium in very deep time, and that's where I get a little uncomfortable. So everything that I did is the past 100,000 years, in which case the rift has split by like, I don't know, the length of this room. Like not a whole lot has changed geologically in this part of the world by then. That said, people do strontium with dinosaurs, for example. And there are absolutely things you can do with that. For example, the amplitude, if you're comparing Triceratops and a hadrosaur from the same site, and one of them has this much variation and this one doesn't, that's still telling you something.
Yeah.
That said, you're not able to like map that out or anything.
On the landscape.
Exactly. Nor do they try to be clear. They also know their limits, but yeah. The oldest anyone's tried to in East Africa is Olduvai Gorge at 1.8 million. Even that makes me a little uncomfortable. That said, I've seen the methods and I don't have a problem with, I don't think they're misinterpreting anything. But yeah, I don't think I would personally do it any older than the late Pleistocene. Just because we don't know. Sure. Yeah.
About the paleoclimate context,
Yeah.
the monsoon was pretty weak during the LGM, right? And the ITCZ was pushed further south.
Yes.
So if you're comparing, like you're saying something was non-migrating in the past, because you're looking at the same location today, but could the migrations have adjusted somewhere else? Like the entire range of migration, the species may have migrated somewhere else because the ITCZ is fluctuating at
Absolutely.
different latitudes.
Yeah, absolutely. So the good news is I'm not the only person doing this now. So we now have studies from, so I'm doing this in southern and western Kenya where I find none or only these extinct species. Since I published that, I now have a colleague, Imai Sige, who's an Ethiopian scholar. He now has results from the basin of southern Ethiopia and from the Afar of Ethiopia that also showed no migration. And then Alex Bertacci is a Yale paleoecologist who did the same thing in Malawi. So I agree, we need to just go up and down the whole rift, but as of right now, we're all getting the same story and we're all kind of at a similar phase in our career too, and we're just sharing our data with each other, like, because I agree, I agree that like hypothetically, like what if the whole migration is in southern Tanzania and we're just not picking it up? Yeah, as of right now, it doesn't look like that. Yeah, it's another question.
About the strontium landscape map.
Yeah,
It looked like, I didn't quite absorb it completely, but it looked like there was abrupt changes.
quite abrupt. Yeah, it's based on it. They map very nicely onto a geological map.
Do you ever find teeth with abrupt change in the, like, in the signal?
Yes.
Rather than a smooth curve, because things were moving linearly across, this species were moving, individuals were moving from one
Yeah.
place to another.
Good.
Expect to see something.
Good question. Let me actually just show a supplemental slide. So keep in mind, the strontium that you're eating doesn't immediately become incorporated into a tooth that day. So based on some work, this shows, for example. So for example, here's the estimated intake here in pink. This is an elephant. This is Nisha. An elephant that Katya drilled back in 2022. Moving from different places. So you do see abrupt changes. In blue is how that translates into the tooth signature. So you see that it is picked up, but only about 50% of the amplitude, which we account for all of that and back model all of that. A bit too complex for like I would need 2 hours to explain all of this. But you are absolutely correct that there are abrupt changes. It's more the strontium signatures are sine curves are the first derivative of the actual change, if that makes sense. Yeah. So for example, if you're going straight from high to low, you're going to see that as a decrease. Yeah. Great question though. I'm happy I had this prepared. Any other questions? Yeah.
Are there, how much is migratoriness polymorphic in the species in Africa today? Like, are there some species that here they migrate and others they don't?
So polymorph. So there's so much variation. Just wildebeest alone. Conveniently, wildebeest do well. If you're an ecologist who didn't read any of my papers yet, or if you just lived in 2023, you would be very, very comfortable in saying that wildebeest either do or do not migrate, but it's completely based on subspecies. There are exclusively non-migratory species, because there's 5 blue wildebeest subspecies, just to take that one species. There are 4 non-migratory subspecies, and then specifically western white-bearded wildebeest are the Serengeti migrants. It's a very clean story that way. We have these are western white-bearded wildebeest, and they are not migrating. So, not only do all these species vary across their ranges, they're also varying through time within what is, I mean, we don't have the genetics and we would never get them, but for all we know, these are literally the ancestors of the modern wildebeest in the Serengeti, or at least part of it, and they've simply just started migrating, and yeah. So yes, migration is very, very polymorphic. Any other questions?
If not, let's thank Kaedan. Training lunch at 1 o'clock in Pacific 3. So, see you guys.